Good morning, students. Today we discuss about the topic plant cell tissue culture. Plant cell tissue cultures mean not only using of the seeds, we are useful into the plant stems, leaves, bark, petiole, and flower buds. And these are development of into the new plant, and this is calling of plant cell tissue culture. The plant cell tissue culture it is first introduced by the Haberland in the year of 1903. So the plant cell tissue cultures, or it is the involving of 5 steps. First one, explant. Second one, sterilization. Third one, preparation in media. Fourth one, inoculation. Fifth one, incubation. So these are involving steps 5 into the plant cell tissue culture. The plant cell tissue culture, the nature plants and the laboratory prepared of the plants was it is the difference between of organization into the plant cell tissue culture. The nature plant, this is having of the sunlight and macro element and micro element. This is taken into the oxygen and releases of carbon dioxide and this is a totally water. It is taken into the plant growth development and reproduction process will be after development of the fruits and develop flowers and fruits after development of new generation into the plant. But the plant cell tissue culture it is development of into the laboratory and these are involving of the 5 steps is there. First one, explanter, sterilization, preparation in media, inoculation and incubation. Explant. Explant means any part of the plant it is taken into the and cutting of the plant. For example, leaf and midrib and stems and flowers. And any plant taken into the open plants is separated of the plant organ. And this is the development of into the genetic material, and it is calling of an explant. The explant it is the first plant. For example, the leaf is taken. For example, the stem is taken, and it is the one petri dish is taken and covered with often cap. So this is calling of the explant, the genetic material organization into the explant, and it is calling of totipotency and cellular totipotency. Next one, sterilization. Sterilizations mean this is the cleaning method and it is the totally cleaning process and it is calling of the sterilized. For example, this is a plant development of into the laboratory and where development of into the different type of bacteria and virus and fungus is development of into the laboratory. So this is a totally cleaning process and this is calling of sterilized, and we are using into the 93 percentage of ethanol and we are using of sterilized method, and the sterilizations we are useful into the glassware sterilization and the culture media sterilization and agar media sterilization, autoclave sterilized, laminar airflow chamber sterilized, and we are useful into the ethanol. So why? Because these are totally disintegrated into the bacteria and virus, and this is calling of sterilization. And next involving of the step it is calling of preparations of in media. So the nature plants we are taken into the directly absorption into the water and solar radiation and light and different type of accepted thing is taken of in plant, but we are prepared into the media and our human being and the man-made it is development of media, and is a plant taken into the organizations, and it is calling of preparations of the media. The media was where different type of medias is there, and white media, ms media, and b6 media, and different type of medias is there. The plant cell tissue culture where suitable media, it is calling of B.Sc media, and white media, MS media. MS media means this is first introduced by the Murashige and Skoob. Both are scientists were identified by the MS media, and the MS media we are useful into the ingredients, micronutrients and macronutrients, and next vitamins, amino acids, and we are useful into the agar method. So this is totally we are taken into the agar, and this is the macro and micronutrients and vitamins, amino acids we are taken into the, and one conical flask were taken off into the agar media. So these are added with of auxins and cytokinins and gibberellins also. So we are useful into the plant roots and stems and leaves. We're total using into the sterilizations of in media. Next one, inoculation. The inoculations mean the separation into the boiling tubes. 10 boiling tubes is taken, and after before we separation into the agar media and taken into the boiling tube and covered with cotton plug, and these are 180 degrees Celsius and 15 minutes it is the incubation process, and this is totally decreases into the bacteria and virus and pathogens is contaminated. And next last one, it is calling of incubation. Incubation we are using into the laminar airflow chamber, and before we are useful into the boiling tubes we are taken into the art hair oven, and after we are using into the culture room, and first we are separation into the the boiling tubes, it is opened with cotton plug, and we are covered with into the first we are using into the spirit and lamp, and these are taken off first explant, it is the funnel, and we are using into the needle, and this is attachment of into the agar media and covered with of cotton plug. So this is totally disintegrated into the after bacteria, virus, and once again this is autoclave method process, and after bacteria virus is contaminated, and these are 7 weeks of in dark and light movement into the organization. The leaf or stems or flower we are totally development of into the roots and stems, are where formations of the first callus is formed. The callus after development of into the roots of the stems and leaves, and total we are plant development of into the boiling tube, and this is the separation into the field, and this is the development of into the new plant, and this is transported into the greenhouse. So this is calling of in plant cell tissue culture. The plant cell tissue culture involving of the steps. The plant cell tissue culture mainly uses into the short period is many plant it is produces into the plant cell tissue culture, and the seasonal fruits, for example the summer season was it is development of into the only watermelon, and mangoes also we are useful into the only winter season, but all seasons we are useful into the plant cell tissue culture, and all plants we are development of into the plant cell tissue culture, vegetables and ornamental, and especially greenery plants we are produces into the tissue culture, and these are useful into the plant cell tissue cultures for synthetic seeds and resistancy plants and pathogen development of into the plants is development of in plant cell tissue culture, and we are useful into the artificial seeds. The artificial seeds we are useful into the transported into the another countries also, where development of in plant cell tissue culture.